All right, everybody, we're going to get right into Psychopharmacology Part II today. The one piece of housekeeping to remind you of is to just take the course survey if you have not already. Otherwise, let's get into it.
Today, we're going to talk a lot about neurotransmitters to begin with, and this is a huge part of psychopharmacology because these are the systems that all of these drugs we're talking about are working on. And then we're going to also talk a little bit about the drugs themselves as we lead into the substance use disorder lecture. So neurotransmitters are chemicals, as we've seen throughout the semester, that we use to communicate between neurons, and they can cause excitation, they can cause inhibition, and then we also saw the second messenger system. So this is really just a throwback way back to the chapter 2 lectures. You should kind of have a picture of this in your head already. The other thing is that there are some subclasses that we didn't really focus on very much, and we're going to talk about quite a few of those today. These we call neuromodulators, which are a class of neurotransmitters, but they are entire groups of neurons that tend to have very broad communication across the brain, and their role is not necessarily to cause the excitation or the inhibition or the activation of second messengers, but oftentimes what they end up doing is modulating the way that these other signals are received. So hence the name neuromodulator. Now, they can have effects on neurons in and of themselves, but part of their job is to modulate ongoing neurotransmission. If we're talking about neurotransmitters, we can't get through that without talking about acetylcholine. Acetylcholine was the very first neurotransmitter discovered. The experiment to prove that neurotransmission existed is something I talked about way back in the very first lecture when we're introducing just the history of physiological psychology and neuroscience. And what I mentioned in that lecture is that there was a long-running argument about whether or not all of the signals in the body and brain were electrical, or whether there was a chemical component, or whether it was some kind of a hybrid system. And we know that some of the systems in the body are chemical, but the communication between body and brain, it was a big question mark, and the communication between parts of the brain was a big question mark. And so what Otto Loewi did in the 1920s was to demonstrate this in a very clever experiment, where he took a piece of the vagus nerve, which is a major nerve running through your spinal cord that controls all kinds of bodily activity, and he took a functioning heart and he put it basically in a big beaker like you see here. And if he stimulated the vagus nerve with electrical stimulation, then he could slow down the heartbeat of this first heart. And this vagus nerve is directly attached to the heart. So in this case, having this piece of the vagus nerve, which one of the things it does is control heartbeat, is naturally going to cause a change if the stimulus is only electrical. But then the clever thing that he did was he put a tube, a hollow tube that was filled with fluid, between one beaker and another containing another heart. And what he found when doing this is that you can actually slow the stimulation of the second heart in the exact same way, proving that this message didn't come from the electrical stimulation that you see here, but it could actually be transmitted through the fluid from one place to the other. So by being able to transfer this fluid, then some sort of a chemical was transferred between the two, showing that there was a chemical messenger system. And it turns out that this chemical messenger that he found was acetylcholine. So as you can see in the brain section here, the acetylcholine system originates from at least 3 different sites, one in the pons, one in the basal forebrain, and one in the medial septum. And these are where the soma or cell bodies are, but the projections you can also see span all throughout the brain. And depending on where these cell bodies originate from, we attribute slightly different roles in different behaviors to each of these 3 different systems. So there are some major roles for REM sleep from these acetylcholine neurons in the pons, whereas the ones in the basal forebrain and the septum are involved in things like learning and memory, and in some cases more generic activation to contribute to both of these. One thing that we're going to talk about a lot today are synthesis pathways. And so you all have open book and open note, so I'm going to go through these pretty quickly, but it's something that you should be familiar with. It's possible that there will be some questions on different either precursors or how things come together for these neurotransmitter systems. So for acetylcholine, the way that this comes together is that we have one molecule called acetyl coenzyme A, and then we have another molecule, choline. And choline ends up being the rate limiting step for creating acetylcholine because we typically get it out of our diet. And these two are combined by an enzyme called choline acetyltransferase, or CHAT. And so what CHAT does is it basically takes this kind of acetyl component of the acetyl coenzyme A, and it transfers it to the choline ion here. So hence choline acetyl transferase. It transfers the acetyl component to the choline, right? So I'm making a funny phrase out of that, but that's exactly where that name comes from. And then this is packaged into vesicles for release by the vesicular acetylcholine transporter. So the previous slide gives us the beginning of the life cycle, but the end of the life cycle for acetylcholine is something that we've already given an example of because we talked about enzymatic degradation way back when we were just first talking about neurotransmission. So acetylcholine is one of these neurotransmitters that's broken down into pieces by the process of enzymatic degradation, and the enzyme that breaks this down is called acetylcholinesterase. And what this does is it basically breaks the bond that we just saw. So we break the choline molecule and the acetate ion into two separate pieces, and then they can be broken down further or recycled, and that's basically the life cycle of the acetylcholine. The acetylcholine system has two major receptors. The first is a nicotinic receptor, and as you might imagine, based on the name, this is the primary receptor that nicotine binds to. What you're going to see for some of these, at least, is that we often find a chemical that's highly used, like nicotine, and then people have gone looking for the receptors, and then we name the receptor and its entire class after this one drug. And the same thing happened for the other class of acetylcholine receptor, a muscarinic receptor, which comes, the name comes from muscarin, which is from poison mushrooms. So these two are found in both the body and brain, but it turns out that the muscarin receptors are slightly more common in the brain and the nicotinic receptors are slightly more common in the periphery, mainly because the nicotinic receptors have a function at neuromuscular junctions. So they have a function in helping motor responses. And for your weird and interesting science fact of the day, this was something that actually, this role for these nicotinic receptors in the neuromuscular junction was actually very problematic during the Civil War because there were reports of people who were having arms and legs amputated, and they were given a drug called curare. And it turns out that curare it works on this acetylcholine system, and it was being used as the first early anesthetic, right? It was putting people to sleep so that they could do surgery. And yet there were a bunch of reports of these soldiers who were saying, I was awake the whole time. I could, everything that you were doing to me, I knew what was going on. And it took until a surgeon himself caught a musket ball to the leg and told everyone, no, this is really true, for them to realize that the drug curare was not actually an anesthetic, right? It's not general anesthesia where it puts you to sleep. It's actually a paralytic. So in fact, this was true the entire time. This very first anesthetic was making it to where all of these soldiers couldn't move, but they were incredibly aware of everything that was happening the entire time. So if you think of a worst nightmare in, kind of early science and medicine history, the curare in the acetylcholine system is definitely one kind of, interesting and crazy case of us not understanding these systems well enough. We've talked a lot about glutamate and GABA this semester as being kind of opposing neurotransmitters, and these are what we put into a category that we call fast neurotransmitters. So glutamate, you've probably heard about all across psychology or neuroscience or other courses, is an excitatory neurotransmitter. So its typical job is to activate neurons and to make them fire action potentials. And this is synthesized, just in the same way that acetylcholine was synthesized using an enzyme called glutaminase from glutamine, which again, most of these things are amino acids that we're getting from our diet and other things like that, and then we convert them into the things we need in the brain and the body. And this is packaged into vesicles by the vesicular glutamate transporter. So, an easy way to remember all of these is that if we're trying to, what do we need to use if we need to transport glutamate into synaptic vesicles? Well, it's the vesicular glutamate transporter, right? So you just reverse it. If I want to transport glutamate into vesicles, I use the vesicular glutamate transporter. That's an easy memory mnemonic, and most of these have something that's along these lines. It's going to be a vesicular something transporter. So that'll be the way that, for all of these, we're going through this life cycle. So that's a great way to remember them. For glutamate, we've also talked a lot about the receptors. We talked a whole lot about NMDA and AMPA receptors when we were talking about learning and memory. And those are really the two primary receptors, but there are also kainate and some metabotropic glutamate receptors that have other functions that can be important. And some of them play roles in the same things we've already been talking about, learning and memory and things like that. But AMPA and NMDA are by far the most common. So as we start to look forward to our sections on substance use, we'll be talking a little bit about drugs that act on each of these receptors. And it turns out that alcohol is an NMDA receptor antagonist, so it comes in and blocks NMDA receptors. And we also talked a little bit about in the last class about these indirect antagonists. And so these are bind on some secondary site. They might bind and they can influence the way the channel functions. And that turns out that PCP is another illegal drug, is an indirect antagonist of NMDA receptors. So, one thing you should be thinking here is, and I'm not going to give you the answer, I'll just pose the question. Maybe one of you can write your thoughts, or a few of you can write your thoughts in the chat back on Canvas, is if alcohol blocks the NMDA receptor, what do we think might happen with memory if we have a lot of alcohol in the system flooding these receptors? We also talked a lot about the AMPA receptors, and these mainly control sodium channels. They're the ones that allow us to unlock the NMDA receptors. And as I said, I've been saying for the past minute here, both of these we already talked about as being important for learning and memory, so I'm not going to belabor the point. We've talked a lot about eliminating glutamate from synapses over the semester because astrocytes need to eliminate this. I've mentioned a few times this idea of glutamate toxicity. So there are a couple of ways that glutamate is eliminated. It can be taken up by various excitatory amino acid transporters. Some of these are on astrocytes or glial cells, and some of them are on neurons. And in many cases, it's also broken down into an amino acid called glutamine by glutamine synthase. So the most important reason that we need to get rid of this is that we can have, if we have a buildup of glutamate, it causes glutamate excitotoxicity. And you'll remember that some of the excitotoxic lesions that we talked about in the methods chapter basically do something like this, where they cause the cells to be so overexcited for such a long period of time that basically the neurons will die. And that's a technique that we can use, but clearly that's not something we want happening in a living brain, especially not all over a brain. So these are the kinds of things that cause long-term brain damage, neurodegeneration, and aging. And so it's really critical that we have these systems to prevent that from happening. GABA receptors, also known as gamma-aminobutyric acid, are the opposite of these glutamate. They're inhibitory neurotransmitters, and GABA and glutamate are probably the two most ubiquitous neurotransmitters in the entire central nervous system. They're all over the place. They have so many functions. And actually, they're related. So what happens is that GABA is broken, is a form of broken down glutamate. So we have glutamate that's broken down by an enzyme called glutamic acid decarboxylase, also called GAD, G-A-D, glutamic acid decarboxylase. And so we get GABA when glutamate plus GAD basically creates GABA. And similar to the glutamatergic system, then we have a transporter to package GABA into vesicles, or this vesicular GABA transporter. One of the things, much like glutamate, is that we have a very critical balance of these neurotransmitters, and a lot of that has to do with the fact that they're so ubiquitous in the brain. So, for example, we need inhibition, and one of the things that we see, especially in epilepsy and other similar disorders where you might have a seizure component to them, is that there's an absence of GABAergic inhibition. So GABA and glutamate are kind of the yin and yang of the brain, and we need them to be in balance. And that balance is clearly critical for all kinds of brain function. And they've always considered to be kind of these polar opposites. Although a fun fact, if you're interested in taking a deep dive into some of my research, I was a part of a group at the NIH that were the first to discover some neurons that are a new class of neuron in the brain that actually co-transmit, which means that they can release both glutamate and GABA. And so we've started to investigate the function of these kind of things. And so they're, an entirely new type of neuron in many ways. So there was a group as I was coming in that, were the first to discover this, and we've expanded on those discoveries since, and proven some of their functions and things like that. So we actually just had a paper out this year showing some of those functions and how to target them. The receptors for GABA are the GABA A and GABA B receptors, at least the dominant ones. And there are so many different drugs that act on these. And typically GABA, unlike glutamate, there are GABA transporters that are used for reuptake, and these are mainly on neurons. So glutamate is kind of the one that has a more special relationship with the glial cells and the astrocytes. There are a lot of drugs, as I said, that target GABA. Benzodiazepines, which are used for kind of anti-anxiety, but also are used in anti-seizure medications. You can see based on what I just said in the previous slide of needing this GABAergic inhibition. But also an interesting thing is that alcohol also acts. So it's an antagonist for glutamate, so it blocks those NMDA receptors, but it's also an agonist for GABA receptors. And then also other barbiturates. So these are in a similar category, often used for anesthesia and things like that. So both of all of the benzodiazepines, the barbiturates, and the alcohol are all GABA agonists. Moving over into monoamines. So these are kind of brain-wide systems, and they often are thought of as falling into the category of kind of broad neuromodulators, which we talked about in the early slides here. There are three different primary monoamines. The first of these is serotonin. The second of these is dopamine, and the third is norepinephrine. Now, you don't need to know these chemical structures, but what I do want to point out here is just how related these are. And it's a little bit less easy to see between serotonin and dopamine and norepinephrine. But if you look at dopamine and norepinephrine, then we really just have this single kind of like hydroxide grouping over here that differentiates these two. So they're related, and we're going to see as we start to talk about synthesis pathways that they're there. But I think you can also see, if you just look for a second, of some of the similarities, where serotonin is the most different of these, but it's also not too far off. So that's something that I think is really important to point out. There's some really important similarities between these. And the reason I say this is because many drugs we're going to talk about end up acting on all three of these because of their similarities. So the monoamines originate back in the brainstem or the midbrain. So if you draw kind of a line up from your spinal cord back to where it touches your head, the back just behind your ears, that's going to be the area where your brainstem starts, and the midbrain is if you just kind of carry forward a little bit. So it's kind of the bottom in the middle of your brain. And these are all really widely distributed networks, as I said a second ago, for neuromodulation. And one of the things that makes these a neuromodulator is the fact that they, we've talked about kind of a typical synapse where a neuron releases a neurotransmitter and another one receives it. But what turns out to be the case for a lot of these monoamines is that they are released, and their releases include something we call volume neurotransmission. So basically, it's not that they're targeting a specific synapse, it's that they might flood an area with many, many synapses, and that means that they can kind of modulate the function of all of these at the same time. And another thing that really classifies these as more of a neuromodulator is they're often co-transmitted with a fast neurotransmitter. For example, neurons in the reward system, a huge number of them co-transmit glutamate and dopamine at the same time. So while you might see that the neuron receiving the signal is, activated by dopamine, there's kind of this lingering question that is probably at the edge of what people are answering right now is how much of this effect is due to dopamine, how much of it is due to glutamate, and in something like substance use disorders or drug addiction, what's the contribution of each of these two substances towards long-term changes in the brain, towards that allostatic load or that kind of repeated insult that we talked about in the last class. And one thing that's common, which is kind of easy for all of these, is that they all the monoamines are transported into vesicles by the vesicular monoamine transporter. So they have a common protein that packages them into vesicles, which should lead you to something or to think, well, how can we do this? And the answer is pretty straightforward and simple, which is that these classes of neurons start out in different places. So we can do this because this protein can be used for all of them because they don't exist in the same neuron. We don't have serotonin, dopamine, and norepinephrine, at least not commonly in the same neuron. So dopamine, we've talked about a few of the systems. We talked a little bit when we talked about the lecture on the motor system about the substantia nigra dopamine, and that being kind of a system used for invigorating movement. When we talked about reward and learning, we talked a little bit about the mesolimbic system. And I think we briefly touched on the mesocortical system, but all of these are three different systems that use the monoamine dopamine. When we think about the role of these in things like psychopathology, we definitely talked about Parkinson's disease as being a disease where dopamine neurons start to degrade and have poorer function. And so low dopamine activity can lead to the motor symptoms we think of for Parkinson's disease. Whereas the opposite end of this, really high dopamine activity, is actually a trait in schizophrenia. And so right in the middle, kind of in the Goldilocks zone here, is where we use dopamine for all the other things that we've already talked about. So normal movement and reward and cognition and all kinds of things like that. So if you see here, you can kind of see these multiple major projections. So the ventral tegmental area has both this kind of meso, meaning kind of middle, so midbrain mesolimbic system, so to many of these kind of limbic structures up here, mesocortical system. And then we also have the nigrostriatal system originating in the substantia nigra, you can see here, and going into the groups of that we collectively call the striatum, which is really this caudate nucleus and putamen kind of area. So that's the very specific projection of that system. Norepinephrine is a system that we talked very little about, but it's a system that's involved in things like arousal and vigilance. Um, and so the place where we talked about this a little bit, but kind of indirectly, is that norepinephrine neurons have a very important role in the autonomic nervous system and kind of generating a stress response and in driving some behaviors that are associated with kind of those sort of fight or flight responses that we talked about in previous lectures. Um, but there's also a second kind of interesting role for norepinephrine in things like attention. And so it seems that, for instance, if you're trying to differentiate between a lot of things going on in an environment, right? If you're in like a noisy room and you're trying to hear, one conversation a few feet away over all the others that are going on, then it turns out that norepinephrine has a really important role in something we think of as like filtering, right? So in trying to take stimuli in the environment and process what's important and separate what's not. And so this is a place where norepinephrine interacts with a lot of the sensory systems we've talked about. One thing that I should say, just to follow up on that too, is that you can imagine how something like vigilance and this filtering are really part of a the same system. Because what is vigilance? Well, if you're walking down a dark alley, you're going to put yourself into a heightened state of arousal, so you can pay attention to new things in the environment, important things, or changing things. So it's not that these two are necessarily different; they very much could be one and the same. So we just saw that the dopamine neurons are kind of in a place right about here, and we're looking at rat brains here, just because that's I should have said that to begin with, but you can see now these neurons in the part of the brain called the locus coeruleus, named for its kind of bluish tint or color that it sometimes shows in human brains. They also have a similar kind of a projection scheme. There are some other places where the norepinephrine projects to. I think the cerebellum is a really nice example here, but these also have pretty wide branching projections, which is something that's going to be common of all these monoamines. So let's look a little bit at the synthesis here. So one thing I really want to point out is that you should know all of these precursors by name, but you don't need to know the chemical structures, as I already said before, and you don't need to know the enzymes. So the enzymes are always going to be these pieces here, tyrosine hydroxylase, dopa decarboxylase, and so all the enzymes pretty much end with A's. But I think it is really important to note, especially because there's a relationship between these, that the way that we get to dopamine is that we have a set of kind of precursors that this builds off of. So we take tyrosine, and then we convert this to L-DOPA, which you should recognize because when we talked a little bit about Parkinson's disease and treatments, L-DOPA is a primary treatment that's been used for Parkinson's disease. And the reason for that is that it is lipid soluble. So it crosses the blood-brain barrier very, very well. Whereas if we tried to just give people an injection of dopamine actually doesn't cross the blood-brain barrier very well. So the L-DOPA is the precursor, and we increase this precursor, and that allows us to get more dopamine because that's the next step in the pathway. And then dopamine is actually using the enzyme dopamine beta hydroxylase is actually what turns into norepinephrine. So this is part of why that chemical relationship is so tight between dopamine and norepinephrine, is because dopamine is kind of like norepinephrine's big brother. We get norepinephrine by breaking down dopamine using this dopamine beta hydroxylase enzyme. Switching back over to the serotonin system has so many roles in the brain, and they're very difficult to study. Some of the people that I've worked with over the years, because I've worked a lot more with the dopamine system, would joke that professors that started studying dopamine got tenure and professors that started studying serotonin got out of academia. But the reason for this is that the serotonin system has always been notoriously complicated. There are at least 14 different classes of receptors, some of them with many subtypes to them, and it turns out to be a very, very muddy kind of a place. And depending on what part of the brain and what receptor and all of these different interactions, you see a very broad range of behaviors. Some of these are related, or that you could at least make a case for them being related, but some of them it's really hard to make that case. So, for instance, mood and pain we've talked about as going together, and I think we think of them intuitively together because you can be kind of hypersensitive to a lot of things when you're in a bad mood, but why those two necessarily have to go together is not always clear. On the other hand, it's a little easier to see the relationship between, sleep and arousal, right? If you have higher or lower levels, you could be kind of in a hypervigilant state or a hyper relaxed state. So those two are a little bit more, I think, a little easier to put together. And then we also have roles in things like eating, which may be direct or indirect when we think about, again, things like mood. Here is a picture of the serotonin neurons and their projections. So we have, multiple different nuclei, so multiple different kind of brain areas that we call the raphe nuclei. There's a dorsal raphe that's up, at least in this case, towards the dorsal end of the brain, and then there's a median raphe, which is more towards the middle of the brain. All of these are part of this grouping that we call the raphe nucleus, or the raphe nuclei, I should say. And again, what you'll see, just to kind of keep a common theme, is that these are very, very widespread projections. So all of these neuromodulators are kind of whole brain encompassing systems that there's almost not a portion of the brain, or not many, where these systems don't target. They're pretty much hitting all of the major areas of the brain through their very widespread projections. Serotonin not being as related to dopamine and norepinephrine has a much simpler pathway where we start out with tryptophan. In this case, that's the precursor. And then we have tryptophan hydroxylase that converts this to 5-hydroxytryptophan, and then finally into 5-hydroxytryptamine, which is also known as 5-HT or serotonin. So there's kind of a three-step process here. And again, you should know these kind of precursors that I've just highlighted here in orange, but you don't necessarily need to know all of the different enzymes that go along this process. And rounding out our talk of the monoamines, there's one monoamine that we consider an atypical monoamine, and this is histamine. So the histamine system, many of you have probably heard of, because I would guess that the large majority of you in the class, or probably all of you, have taken an antihistamine at some point when you've had allergies. And we talked a little bit about this in the pain and the itch system. So the histamine system mainly originates in the hypothalamus, and you might imagine, if you've ever taken Benadryl, the histamine system has a role in wakefulness. So it has these roles in itch and things like that out in the body, but it also in the brain has very important roles in wakefulness. So if you've taken Benadryl, you get very sleepy, or at least many people do. There are some people that are kind of immune to those side effects, and some people that are really hypersensitive. For example, I tend to be kind of hypersensitive to Benadryl. If I take it, I'm going to be asleep within the next couple of hours. And antihistamines are particularly good at causing drowsiness, but the trick with some of the newer ones is that they don't cross the blood-brain barrier. So that's helps for things like Claritin and other things like that to prevent some of these negative side effects. And if you remember what we talked about in in just the last lecture, if we're looking at this margin of safety, of course, if we can do something like that, whoever's doing the drug development can prevent these from crossing the blood-brain barrier and prevent this really negative side effect of drowsiness, then you can drastically increase the margin of safety. So rarely, but sometimes work that I've done is on human brains that come from brain banks. And, one of the things I will never forget is that I was being shown in order to kind of evaluate what would happen, the process for how a brain was taken in from the medical examiner and basically collected and how the entire processing went. And I happened to go in, and I was still a very young researcher, and went into this National Institute of Health suite where they had just taken a brain in from the medical examiner in Virginia, and it turned out that the young woman who had died was exactly my age, which is a scary thing. And the scarier part was the way that she had died was that she had been out drinking at a barbecue and had been itchy because of the grass and had taken Benadryl. And so the combination of those two is, of course, a deadly combination, and that's something where the margin of safety should become, very clear. So in some ways, I tell this story to tell everyone never to take Benadryl and drink alcohol. It's a terrible combination, and I think a lot of people don't realize how easily some of these interactions can happen. But that is also something that points to the reason for these needs for these big margins of safety, because something like that is it's a situation that many people could enter into, where you would think that these two things are a good idea, but maybe just not together. So that's the sort of thing the FDA and others have to worry about when we're thinking about psychopharmacology. Anyway, walking away from that story for a minute, which is probably not the best 2020 lift you up story, but is a very important one for the realistic implications of what we're talking about, is that if we're trying to produce histamine, then we get this from a chemical called histidine through the enzyme histidine decarboxylase. Another category that we haven't talked very much about are neuropeptides, and I bring them up every now and again, but we haven't really spent much time on them. So just by comparison, neuropeptides and neurotransmitters in the end have the same function. They're both ways that we can signal in the brain. But neuropeptides are kind of like larger groupings of amino acids. So they typically are 2 or more amino acids that have peptide bonds, which means they're really large. And because they're larger, it means it's more difficult to package them into vesicles and do all kinds of things like that. Whereas neurotransmitters tend to be small. So this means that pretty much every stage of the process, even these precursor molecules and things like that, need to be bigger. They take more time, and so the neuropeptides have an entire packaging pipeline, whereas a lot of the neurotransmitters, we've seen that they're produced. Many of the pieces are produced at the soma, but they can really be packaged and they're kind of malleable at the synapse, right? Where they can be repackaged or they can be put back together from pieces if there is some sort of degradation. And it's very easy for the brain to just work with these. They're maybe a little bit more like Play-Doh, a little softer and easier to work with. The neuropeptides are more like a hard clay that you need to get in place and get perfect. And then it's a little harder to work with. Neuropeptides, another thing is they come from these vesicles called dense core vesicles, and so they can be released from all parts of the terminal, whereas neurotransmitters are more likely at the synapse. So neuropeptides are not, kind of like these monoamines, they're not a traditional signaling mechanism. And often the neuropeptides are co-released with the neurotransmitter, and they take a lot of energy. So it's usually not a single action potential is releasing these peptides. It's often the case that like a string of action potentials or something else triggers these dense core vesicles that have kind of one neuropeptide packaged to release and to bind. And for all the neuropeptides, something that's common is that they have no reuptake mechanism. Whereas we've seen that the neurotransmitters, some have enzymatic degradation, but for the most part, a lot of them are recycled and reused. And so this is never the case with neuropeptides. They're created, they're packaged, and they're kind of a one-time use, but they tend to have very important jobs. One really important class of neuropeptides are the endogenous opioids. So we've talked a little bit about the endogenous opioid system when we talked about pain, and there are three major classes: the mu opioid receptors, which have kind of primary roles in analgesia or anti-nociception, so they work to inhibit pain signals. And then we have delta opioid receptors, which play somewhat of a similar role. They can have analgesic effects, but they also have roles in things like mood as well. And then the last are kappa opioid receptors, and these tend to be ones that are associated with aversion. Even though they can have some roles in anti-nociception and analgesia, they tend to be kind of the opposite in many ways of these mu opioid receptors, even though this diagram here shows that, yes, of course, they play some roles in analgesia and things like that, depending on the system. But opioid reward is typically, in something like drug abuse, is typically driven by the mu opioid receptor, whereas the opposite, the major withdrawal symptoms and things like that, are typically driven by the kappa opioid receptor. These are kind of the opposite pieces of that homeostatic or allostatic load that we've been talking about. One tends to drive things towards reward, the other one tends to drive things towards aversion. Lipid neurotransmitters are an interesting class that we mentioned a little bit in a very early lecture, but I'm not even certain we ever called them lipid neurotransmitters. So the primary lipid neurotransmitters are in the endocannabinoid family. So again, we have talked about cannabinoids and endocannabinoids. So these, like many of the other things we talked about, are named from cannabis. So again, a system of neurotransmitters or receptors are named after the drug that we're studying to bind to them. And so endocannabinoids, in thinking about drug abuse, are basically responsible. It's the system that's affected by marijuana or THC, which is tetrahydrocannabinol. And so you can see again in the name here in the cannabinol, right? This is coming from cannabinoid. This is where kind of all of these pieces come together. And these tend to have some positive effects, so they can also have roles in pain relief. In fact, they're becoming, in some cases, kind of a preferred mechanism for pain relief, especially in chronic pain, in things like cancer patients and things like that, because they don't have quite the same abuse liability, but they do have abuse liability. So for those of you thinking about our very recent New Jersey legislation, there's still major ups and downs to the legality of marijuana or any sort of cannabis products. To give you my shortened version of this, I think that there are major problems with the criminal justice system related to marijuana in the country, and I think that that's the positive side of this. And I think that from the perspective of a drug addiction researcher, that the negative effects of marijuana and other THC containing products are often downplayed as a part of that, the argument on one side. And so I think that there's major pros and cons here, and it's something that if you are interested in, you should look at both sides with an open mind, which is the same thing I tell everyone as a scientist, is that you should look at the sociocultural perspectives, you should look at the drug abuse literature and all these kind of things. Anyway, there are also some major negative effects, and so we've talked about some of these systems across a lot of different lectures, and Things like impaired concentration, memory problems, and altered perception are not necessarily things that you want to be pushing long term. So this is where we see these positive use cases for things like analgesia, but then if you add these to things like impaired concentration and memory, then there's some balance there that doesn't necessarily make it a clear-cut case for what somebody should do from a legal perspective. So anyway, I'll get off my soapbox. So something you should think about in us calling these lipid neurotransmitters is that, so many important parts of the brain are made with these lipid bilayers. The cell bodies are made up of lipid layers, the synaptic vesicles. And so one of the interesting things about endocannabinoids is that they're synthesized on demand. And the reason for this is that they are really hard to store and contain. So we basically create these when we need them, and then we break them down immediately. And then create more when we need more. There are two types of receptors, the CB1 and CB2 receptor that are the primary cannabis receptors. And these tend to be found at presynaptic terminals. So this means that they tend to modulate the release of other neurotransmitters, and that's their primary job. So the presynaptic terminal is the one that's typically sending a message, and remember the postsynaptic terminal is the one receiving it. So when you have something acting on the presynaptic terminal, it's affecting how much neurotransmitter that cell is releasing from its axon or things like that. So for the endocannabinoids, the endogenous chemical that is used in the system is one called anandamide. And for most of these, for the CB1 receptor at least, these open up potassium channels, which if you think about all the way back to us talking about the action potential, one of the roles of potassium channels is that after hyperpolarization. So if we have, a typical action potential that kind of looks something like this, then the place where we were talking about, if we draw our kind of baseline level here, the potassium channel is in this zone. That's a bad color. In this, got to go back to something you can see, is in this zone here. It's still not the best, but at least you get the idea. So these after hyperpolarizations then can change the duration of the action potential because we need to get all the way back to our resting membrane potential here before we can fire another action potential. So this can cause action potentials to fire more slowly. If you increase the after hyperpolarization, or if you decrease it, then you can actually cause neurons to be able to fire faster. But also having these potassium channels the entire time open, because remember they open somewhere around here, can also change in some ways the properties of the action potential itself up in this range. So there's a lot of broad effects that these can have on the way that our brain signals, and they're not always a positive effect. One of the reasons that change in it is important for things like memory formation is that if you had this happen at the level of, let's say, AMPA receptors, you might have smaller action potential or fewer action potentials. You might have, more or less neurotransmitter release, which can then change memory formation. So one of those negative side effects is that we change neural signaling, and that means that we don't have as many NMDA receptors kind of becoming unlocked as we saw was necessary for memory formation. And then, of course, we start to see deficits in memory formation. And then coming back to kind of one of our major themes here, anandamide is broken down by this chemical called fatty acid amide hydrolase. So that the starting life cycle, we produce these endocannabinoids when we need them. And this is, I haven't asked you to know kind of the starting pathway, but they're produced on demand. And then anandamide is broken down in the end by an enzyme. So it's, and again, another case of enzymatic degradation. So in trying to wrap some of these things up for today, and we're going to talk a lot about some of these systems down the road in the next lecture on drug addiction, when we talk about specific drugs and what chemical systems they interact with. You've seen a few hints of that today, especially when we talked about alcohol and opioids, and I think you can imagine some of them. But just to give you some tables, like I do sometimes, you can see some of the CNS functions. So these things would be really good to know. If there are any peripheral nervous system functions, those would be good to know. And then I've kind of categorized these to where these fast neurotransmitters, glutamate and GABA, are together. And then some of the kind of more neuromodulatory things here, acetylcholine, dopamine, norepinephrine are here. And rounding these out, this should give you a pretty strong idea of just some of the basic rules for each of these. So this table is more of a summary. It's less important than some of the other tables I've given you in the past, but it would be a good reference for you just trying to go back and study and create some guides for remembering kind of the major functional roles, and then remembering, of course, which neurotransmitter should be on your list to think about from the second part of this lecture. So with that, I will just tell you, we have one more lecture left on drug addiction or substance use disorders. There will be a very, very important hint for those of something you should remember for the final quiz. But again, please check the announcements for the past 2 sections. And if you have any questions, pop into office hours. And we're almost done, so hang in there. Catch you next time.